So today we're going to continue our series on structural analysis of proteins. So as we had last time, we had the 1ZNI structure, which is insulin in a dimer form. So here you have insulin with ions right here. So this would be sodium, and this would be probably zinc. Yes, zinc. So right away, just before we keep on going, you'll see that sometimes when you open up protein structures, you don't just have the ions or just the main backbone, but sometimes you have key amino acids showing. Usually these are amino acids highlighted to show that they're actually interacting in a particular way. Usually there's a lot of interaction, but interaction with something outside, let's say a ligand or an ion, can be showed sometimes to highlight something maybe special about the protein. So to move around, I use a Mac. So to move around, you have your touchpad. So right now if I use single finger, you can see I'm just moving my cursor around. If I use two fingers, what's going to happen is I'm going to rotate. So the file containing the position of these amino acids actually has an origin. So basically when you move around with your two fingers, what's happening is actually you're moving or rotating around that center. So you can play around with it. If you have three fingers, what's going to happen is you can actually translate. What might happen if you go too fast, like on my computer, so you might actually have the windows moving. If you move quite slowly, there's other ways to do it of course, but if you move quite slowly, you can still have that nice translation. Again, two fingers, you can actually zoom in again, so instead of just moving them around with the same distance between your fingers, you can actually increase the distance between both fingers to actually zoom in and out. You can actually combine this also with a rotation. Okay, just going to put this back in position. Now, if you want to use the keyboard and the notepad, what you can use is function, so the FN button. FN and one finger with a click can allow you to rotate like we had with two fingers. Okay? So that nice circle can indicate that you're in rotation mode. If you press Control and a simple finger, what's going to happen is your click's going to create a box, that green box you see right here. And this is a nice selection box. What you can do is actually click with one finger, and then with the second finger, while the first one is being pressed, is actually scrolling down. So what I said is, you go Control, you click, you maintain that finger on the notepad, so the click is maintained, but at the same time you take another finger and just drag it along to actually make a nice window. So these selection boxes can be very useful, as sometimes you might want to select a simple amino acid, or a location, or say an atom itself, or maybe an ion. But sometimes you want to get large regions, so you just Control, click, and drag, and make a nice big selection. If you want to click on a single unit, a single position in the PDB file, you just click Control, and just click once with your notepad. So once again, to make a window with Control, click, drag, that's for big selection, and Control, click allows you to take one selection, and again function allows you to rotate. Okay, so Option here, with the equivalent of Alt on Windows, actually allows you to translate on the plane that's facing you, right? You're not moving on the Z-axis, you're at X and the Y. So this can be very useful if you want to focus, if you're really zoomed in and you just want to move around, you can press Option or Alt and just click around it and move around, right? And then you can click with Control click and Control click elsewhere if you want to deselect it. Maybe if you want to have multiple selection, but that are really local or very far apart at the same time, you can, instead of controlling, and if you click there, so let's say Control click here and you Control click here, you see camera makes a decision on only one of them. But if you want to select both, what you do, you click Control, you click the first location, then what you can do is press on Shift. Once you're pressing Shift, camera knows that it wants to keep that location in whatever you're going to click next. So let's say I want to keep this, and then I want to keep this, and then I want to keep this. So it's selecting all 4 locations. This can actually be applied elsewhere, as we will see later. For example, in an amino acid sequence, there's different motifs in the long amino acid sequence of interest. You can actually select only them, even if they're separated by multiple amino acids. So if we zoom out again with 2 fingers, just scrolling out and in and out, you can just deselect by just Control and clicking in the dark area in the back. Okay, and there's one last thing. There's zooming in also with Command and dragging. So if you drag your finger on the notepad while pressing on Command, you can drag on the middle of your screen. So that's a little bit different from what we saw before with the rotation. Now we're just directly moving in the center of your screen. Okay, so that's basically moving around. Get comfortable with it. You'll be using it quite a lot in these tutorials. So now, once that's done, we're going to start exploring the menu right here. There's actually ways to do what is called commands, but we'll go over that maybe later. But the most important thing is basically understanding these menus right here. So you can have the files. So if you want to import, like we said, you can fetch by ID. There's different databases. We'll almost probably actually always going to use PDB, but there's other ones. Maybe there's a particular situation where you're going to actually have them. You can set a download directory in your computer, right? So you're always going to download from there. You can download from your files, or you can download them to save them actually in different files. So there's actually Mol2. There's actually export the scene. So the scene is going to be the JPEG or the bitmap. That's going to be simply the picture out here. Publish going to have different nicer variants, so a little bit cleaner, better for publication. And PDB, of course, and Mol2. Mol2 is a different type of file. So if we export scene, there's actually STL, very interestingly, is used for 3D printing. Okay, so there's different actually. Our Chimera supports multiple file types, so this can be very interesting depending on your use. Okay, so this is for file. In select, you're going to have all your options to get even more precise with your selection. So the basic selection that we've seen is now it's Control click or Control box to get something a little bit bigger. But you can actually select by chain. So here we have 4 chain. Why 4? Well, it's because there's actually been a crystallization problem, or not problem, just the PDB is not containing this part right here. We'll see how to actually put that back a little bit later, but right now what we're going to do is actually basically consider this as a tetramer. So actually 4 different sequences, because for Chimera there's actually 4 continuous structures in the PDB. So Chimera doesn't know that these are actually the same protein. We'll have to edit that a little bit later. So we have those 2 insulins that are actually broken in 2, right? So what we'll do, that's interesting, very interesting actually. I wonder if this is part of the rest of the insulin or something else, whatever. So once this is done, you can select by chain, so A. Okay, so in the PDB, probably these is A, B, C, D. So selecting A, selecting B. So we see that actually the ions are actually associated with some of the sequence, and it can go on to C and others. Okay. Chemistry can be sometimes very useful. Elements, I've never used them particularly, but let's say you're working with, you want to change all the colors of your phosphorus, for example. Well, that could be a great option because it could bypass any manual intervention. So let's say you want all your oxygen orange. Well, just pressing on that will select all your oxygen. Obviously, because there's oxygen in all amino acids, and everything, not this, but all the background are actually being selected because this is unspecific. So it just says, well, for that position, there's oxygen right here, so I'll select everything in there. But actually, I'll see later if you can actually transform this to show only the atoms, and that could be very useful. So chemistry elements, functional groups, that can be slightly more useful. Actually, all of these are very useful. Just for my own personal purposes, for drug discovery, drug editing, these can be very, 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 very useful. So let's say you have complex ligands, you have multiple of them in different families. You can all the pyrimidines, for example, or simply all the aldehydes. We see here that there's different amines, so there's pretty much all the important organic groups. So that can be very useful for getting gross selection of everything containing these. And the ions in actually different ionic states, so that can be very useful for different crystallization states. Okay, so residues is similar to what we had here in functional groups, except they're highly focused, of course, with the amino acids. So you can select any of these. There's non-standard right here. So everything that's in there that's not supposed to be an ion, but looks more complicated. So there's zinc, chloride. So they're not amino acids, but they're key units. So they're individual units in the PDB. And OHs, which are, of course, water. And there's different strategies. So let's say you want to find everything that's aromatic in your protein, and that could be very interesting depending on your drug target. You could look for everything polar, everything negative, and basically everything's going to be selected that fits this category inside the PDB. And structure, of course, so everything that's backbone, ions, ligands. Ligands can be very useful when you only have the ligands. Actually, in this case, if you select ligands, there's nothing. So Chimera is actually pretty smart. It knows that zinc is not, it's actually by name, but it's not a ligand per se. It depends on your definition. Okay, and there's solvent. Let's say you hop on a PDB. There's water in there, right here. So you see, there's a single water. It's been detected. Well, let's say there's a lot of water around it. You want to delete it right away. As you will see later, you can just add a bond and then delete it. Okay. Then sequence. So as I said before, okay, you can have the sequence. Okay. So let's say there's all the AF. Okay, it doesn't look like there's any AFs in here, but you could do that. So find a little motif. Let's say binding sequence or recognition sequence. You can hop right away, check if it's in there through sequence. Atom specifier, zone. Zone can be very useful if you want. Let's say I want to have a zone. I want to select every atom or every amino acid that's in a zone of 5 angstrom. Let's say everything that's 5 angstrom. This zinc. So it's going to calculate the centroid of this zinc, and you can select everything that's below 5 angstrom, everything that's above. So here you see it's selected everything that's 5 angstrom. So here the backbone is very far, but as you can see later, you can actually here action atom show. And probably this amino acid or something in here is 5 angstrom, so it's selecting the whole backbone. Okay? So that can be very useful to find the general region, general amino acid that are binding or interacting with some type of region. So zone. Clear selection, these are, I've never used them personally, but they can actually change your selection type. So we've just undone, okay? And you're just going to toggle between back and back, okay? So here is the interesting stuff, the very interesting stuff. So you can then modify things on different degrees of complexity. Let's say you can modify things with their atoms or bonds, then the ribbon, then the surface. We'll go over what a surface is. And then here you're allowed to change colors, add labels. So let's say you want to label, let's say you have a zinc at this position, you want to label it, or even better, alanine at position 13. That's very important. You want to show where it is to refer to your description or your paper. You could do that through labels. There's different ways actually to do it, but label is one of them. And focus and set the pivot. Okay, you can actually write to PDB. So if you want to modify something without saving the whole session, you can actually modify the PDB. So let's say you add an oxygen at X position, while the PDB is going to be changed in accordance to whatever you change in here. So let's actually start playing with this. Say we select here. Well, what is under there? if I let my cursor, you can actually see that we have leucine 17D. D stands for the sequence number. So when we have the sequence here, chain, it's a chain number, sorry. So it's under here and D. So there's A, B, C, and D. So this was A, B, C, and this is D. So let's see what's the orientation of our amino acid. You can show it. Right? So through action, atom / bond, show. Okay. Let's say we don't want to see it, we can just do the opposite. Just hide. Right? So we can change different things here. Backbones only, side and chain, base. Right? It's going to do the same thing in this case because there's nothing too complex. Then you can change the way we see it, which is slightly more interesting. You can have it in stick, which is actually the way we see it right now. Bond stick. Okay. Sphere. So basically the volume they're occupying in space. Why? So here it's very minimalistic. So here, right now, okay, this is a great example. We're off focus. What we can do, simply press on Option and scroll here. Remember, Option is Alt on other keyboards. Okay, so just select it, and then we can change once again. Oh, there's actually the width you can change. So if you want it thicker, okay. It's nothing comparable to sticks, but it can be slightly bigger. And if you have nucleotide objects, so that's when we're going to go over DNA. DNA can be interpreted in different ways, and a camera can adapt itself to it. Okay. If we have an aromatic ring, actually, let's find one. So here, let's say you're being asked, or you're trying to find a particular amino acid, and you simply don't want to scroll over and check where it is. So there's many ways you can do it. My personal advice to you, so let's say we're going to keep it like that, is to find it in the sequence, right? Because usually the amino acid of interest has a number, and you know its position. So one way to do it is go into favorites and into sequence. So here we have multiple sequences. It's pretty rare. Usually you have two if you have a big dimer. Sometimes you even just have one. So let's say we know we're having chain D. So let's go on chain D, and we have our sequence right here. So here we have our single amino acid here. In File, you can load different things, change the colors, save as. But usually for alignments, you would use different programs, so that's not very important. Here you have multiple other ones. You can do alignments, realign. You can add sequences to this. You can insert all gap columns, and you can actually find subsequences. Let's say I want to find this. I have a really long sequence. I want to find where I can find LIL. I can apply, and it finds it for me. You get ProCite. ProCite is a tool that finds different site of particular types. Okay. Well, ProCite, we can actually hop on here. So ProCite is a tool that allows you to find different things. So ProCite has its own code. So let's actually use zinc finger. Okay, it actually doesn't recognize it. Okay, let's try once again. Copy this one, let's say. Let's drop this down. Oh, okay, so this was actually modeled, but there was nothing found in there. But it can be very useful. Okay. So this is done. This is done. You can have modeler, which we'll go over. It's actually a way to refine it to complete the loops that we have empty association structure. Multiple of these options are going to be seen later. Trees, if you have an evolution tree, we have one sequence, so there would be nothing there, so there's no options. Preferences, structure, header, regions, appearance. No. Multiple options, we won't go too much details, it's not that important. As I said, usually you don't use Chimera itself to do alignments, but you can do some alignments. You can actually you can find annotations, and I did the sequence name, but we'll actually just use it to define position. So let's say we want to find an aromatic. We're pretty lucky. The first one is. So as I said, you can select by clicking, and you can actually select multiple positions that are far apart through the option of using Shift. So when I press and click Shift, I can select both of these amino acids. I want to reselect just this one, just click on this one. If you want to have a large sequence, you can just click and drag. So once again, just clicking. Let's say I'm really interested in this proline and in this glycine. Perfect, that was done through Shift. I'm really interested in this phenylalanine. I can also Shift and click. Okay, so now I'm just interested in two phenylalanines. I just drop the Shift and just select it again. Now that phenylalanine is selected, let's find it. So it's going to be underlined by our wonderful green surrounding right here. So we know that phenylalanine is right here. Again, want to show it. Action, atom, show. Whoa, perfect. So now if we want to edit our rings, we just can have it here. Just can have a full circle. Okay. Oh, there's actually multiple. There's nice histidine right here. There's actually multiple designs. So that's going to allow you to make nice pictures. The ribbon can be done similarly. You can hide the ribbon, in this case, or show the ribbon. You can have a surface. So the surface is actually more useful if I select a whole sequence. So let's use here, and action, surface, and show. So it's going to calculate the surface, so the electromagnetic surface. So here you can see it's taking a lot more space. This can also be colored red. It's actually changing everything of the selection. So let's dive in more into colors. Okay, let's say we're going to use this sequence and we want to change its color. Action, color, we have a vast array of color, but we also have coloring by heteroatom, which is usually done by element, by editor. Editor offers you actually to move in the RGB strain. Okay. More importantly, for color, it's going to be from all options. So here you can color and apply to different things in a specific way. It's nicely visual also. To change a background, you can do so. So turn back. You want to change all of the above. So, and not only this was selected, but if you had multiple things, everything, regardless of what it was, will be selected. Atoms bond, ribbon. See that everything changes accordingly, but always from selection. Okay? So if you want to change things you selected before using Control, Shift, or different techniques using the sequence, using specific techniques that we'll see also later, and you can change them individually in different ways, but also from this editor, it's a lot easier. Then you can hop onto the editor or by element or by heteroatom. Every option is here. You can also do a lot more if you want by show all colors. Okay? But preferably, usually it's protein imaging. It's nothing too fancy. But if you're really fan of, let's say, goldenrod, or very, 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 very fan of olive drab, well, you have these options also, aside from these more classical colors. Okay, this looks pretty much like a mess, but we'll change that very soon. Before we leave, so once again, right to list, right to inspect. If you inspect, you can actually see all the properties that you have right here. So you have in the sequence selected, you have one molecule per model, one residue that is selected right now. There's 9 atoms and 8 bonds. So in this position, if we show everything, there's actually all these properties. So there's one molecule, there's one residue, that's one residue. There's 9 atoms in there. and 8 bonds. Okay, and then you have the different properties associated to that position if you're ultra awesome editor. Okay. Zoom out a little bit. Okay, look at our monster. Okay. Then we can approach the last thing of tonight. So tonight we'll go over this. It's the presets. Presets can actually be changed in the code or in actually through here to add custom presets. They're very simple, can offer you a wide range of representations soon after hopping on Chimera and looking at a new protein to either analyze or simply to make your final picture. So through ribbons, it does two things, simply ribbons, and also adds a nice rainbow coloring, which is very nice. All atoms, like I said, it's pretty personally ugly, so will rarely find itself on a publication, but can be very useful to mark different amino acids positioned in space, but also individual atoms, right? So if we hop back on into a selection tool through chemistry and through, let's say, amines, there's none. However, if you look through elements and all carbons, we see that all the carbons here are selected. So you can go around and play around with all these presets accompanied with all these other options. There's also hydrophobicity surface, so now it's red for hydrophobic regions and blue for hydrophilic. Right? So here you see it's of course charged. It's actually being binding here. It's actually pretty weird in their work. It seems that this has this conformation right here. It's outside, but maybe an error of Chimera or just an error of the PDB itself. You can watch out for these. First time I actually see this, but you can see the ions are actually nicely fitting. And you can see, oh, probably good jug binding pocket right here. So this can be very nice. Going back to surface, you can play with its transparency. You can completely hide it. You can change also to solid, which it is right now. You can change to mesh. So it keeps its coloring, but it's in a mesh. Can be nice depending on what type of picture you want to do. Dot, which is very, very scarce, but gives you a nice sense of where it is actually in space. And let's be back in solid. You can then change its transparency, let's say to 40%. Or no, actually, let's go to 70. Okay, so now what we see is actually transparency of the surface. Okay, so you can play around, toggle with it a little bit. Okay. Okay. It's actually pretty weird looking at it. It's actually not calculating for everything. It's like if these amino acids, these atoms weren't taken into account. But usually it should work. It's actually the way I actually do it all the time. So once this is done, you can actually hide the surface if you don't want to see it again. Okay, I understand. So Chimera created two partial objects here, and for some reason, but you can understand that, first of all, calculating surfaces can be actually quite intensively computed. Actually, when you have a really, really big structure, it can actually crash your Chimera session. Okay, so let's just change the preset. Ribbons, perfect. Let's make it disappear. Perfect. And there's other presets, so you can accompany. These are a bit thicker, right? You can change the way that a ribbon's being represented. You can flat ribbon, right? So there's multiple ways that Chimera can allow you to design your picture. So in the next video, we'll go over this long sequence of tools. So these are actually the strong point of Chimera, allowing you to do multiple things to your protein, different analysis, such as distance calculation, changing amino acid, filling gaps like these, adding names, calculating the hydrophobicity if it could be actually stuck in the membrane. So we'll go over these tools in the next video. Hope you guys liked it. Have a good night. Bye bye.